Okay, so now you're more or less fit to conduct your own single-cell RNA sequencing data analysis with scanpy. Okay, what now? When you want to have your project organized, I would like to suggest you the following structure. You have seen it already in the course material of day 1 and 2, and you probably have wondered, why do I have so many empty folders? So what I think about these folders is that, for one, you can have code, something like scripts, which are not notebooks. For example, when you want to run something in R, what you typically don't want to do in a notebook, then you can save it as a script in this code folder. Data is usually all H5AD files that you created during your analysis. In the figures folder, you can have all the figures that you created in the course of your analysis. Notebooks is notebook files, so I piped in Jupyter Notebooks. Reports can be anything that you can compile from your analysis. For example, presentations, written reports, manuscript drafts that could go to reports. And finally, you have tables, such as lists of differentially expressed genes or anything else that you can save as a table. On the notebook side, you can have such a structure. For example, when you did the pre-processing, once you're sure about your pre-processing, you potentially don't want to touch it again. So you save it as a notebook, and at the end of this notebook, you save your end data object. That is the final result of your pre-processing, so you don't have to open up the pre-processing steps again. Once you have done the pre-processing, you can have a notebook for a cluster annotation, so where everything that you do is about how do I name my clusters. Then you could have a notebook where you only look at velocities, so like the RNA velocity analysis we did in the last session. Or you can have a notebook where you only look at pseudotime and branchings. And finally, I have sometimes, or most of the time, a notebook where I do lookups, so which is unsorted requests that I usually get from my collaborators. Of course, this list is not closed, so you can easily add notebooks for differential expression analysis, gene set enrichment analysis, or other special topics. So, to have it more as an overview, pre-processing is everything that comes from loading data, merging data, quality control, normalization, batch effect correction. Cluster annotation is annotating cell types, then we have RNA velocities, we have pseudotime, we have unsorted requests, etc., etc. With this kind of structure, it helps me to get organized with the notebooks, and it helps me to keep the notebooks small. So I know what is happening in each of those notebooks, and the notebooks don't get too large, because what usually happens when you have too large notebooks is that they clutter your memory, they open slowly, they have a really sluggish behavior, and notebooks that become larger than 100 megabytes cannot be saved. So that's a lot of motivation to keep your notebooks small and organized. So once you want to look up something, you don't have to look through 5 different notebooks to find what you're looking for. From the data structure, I suggest that you have several versions of your data object. So for example, you can keep an unfiltered version, just that's where you have merged all your samples. You could keep a normalized version, a batch corrected version, and an annotated version. The last three, normalized, batch corrected, annotated, you can also save in one object using the different layers. For the unfiltered version, I would suggest to keep it as a separate notebook, so that you can always go back to this notebook once you decided that you want to change your normalization or batch effect correction or your pre-processing, so that you can easily go back to this notebook without having to load all the data again. So that's what I would like to say about project organization. Now the part comes with reproducibility. This is a thing that it's becoming more and more important, also with respect of code sharing and data sharing. Some journals, especially the high-impact journals, actually require you to share your code. It does not suffice anymore to say, okay, I can share my code upon request. You have to share it. You have to share your notebooks. You have to share the code. So when it comes of closing a project, that's the latest time point where you should clean up your notebook. Make sure that your notebook can run from top to bottom without errors. Especially if you try out things and try different things, it's very easy to just go up and try something and go down again and go up again and go down again in your notebooks. And this is a really nice source of error that in the end will really cause confusion, especially if you try to run things or try to look again at your notebook and see what you did at some point, and jumping from bottom to top cells and changing things will lead to this confusion because you can't really reproduce what you did at some point. Furthermore, it's helpful to add dates to figure files and tables, because you will create multiple versions of certain figures. And alongside with your documentation that you should do for each day, you could revisit your personal notebook and check what did you do on which date and what did you try out on which date, and then immediately compare the figures that you create. And it helps to also find figures again that you created at some point. Furthermore, once you've cleaned up everything and you wait for the revision of your project, and it's probably a half a year or a year later, and you want to look back at what you did like 6 months ago or a year ago, things get nasty because package versions have changed, because you have a new Python version or you updated your operating system, and then it gets difficult to reproduce results. So first of all, I highly recommend to work with environments, especially when you work with the Python Conda package manager. You can create environments. So for example, then you create for one particular scanpy version, you create an environment, you fix all the packages that you have there, and once you want to try out a new package, you create a new environment, and then you can safely update packages there and try things out. Otherwise, don't update. Update only if really necessary. And if you update, make sure it doesn't change result, because things might be different across different package versions. Or you can create a Docker container where you also fixed all the versions, and that has the advantage that once you have to revisit a project that has been off your desk for several years and you most certainly have updated and moved on with your package versions, you still have some kind of a container where you can actually easily recover all the old package versions that you had. I already mentioned this part about Python environments, which are supported by Anaconda. What else? Whenever things do not work, you have to report package versions anyway. You have to report them for publication. So this is something that's really, 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 really important to actually document this. Okay. Again, when it comes to organization of projects, you have to find a style that works for you. The suggestions that I made, you can take it or leave it. In the end, when you try to come up with your own project organization, try to think of yourself in a year from now who tries to recover what you did a year ago, and try to think in advance how you would try to recover things or how you can make your life easier in a year from now.